Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Miss Purnima Bhanot. She's an associate professor at Rutgers, and we're going to talk about malaria, the biology of the malaria parasite. So, Purnima, thanks for coming.
Thank you for having me.
Yeah, for listeners that may not know much about malaria or think about it, what is it, and how many people does it affect?
So malaria is actually a really old disease. It's been around with humans for as long as we have known agriculture. In a good year, which means more recently, there have been more than 200 million cases of malaria each year. And each year that results in the death of over 400,000 people. As recently as 10 years ago, these numbers were almost double of what they are currently. But even with that progress, you can tell this is a disease that takes a huge toll on human life, mostly in sub-Saharan Africa, and mostly of children below the age of 5.
Right. So when someone gets malaria, what happens to them, and how fatal is it?
So in terms of what happens, like the kinds of symptoms people experience with malaria, it's generally high fever and chills. In severe cases of the disease, there is anemia. If pregnant women get malaria, they can often miscarry or have babies that have low birth weight. In terms of the reason why especially kids die of malaria is because the disease can very rapidly lead to coma. So, for example, there are many descriptions of children, seemingly healthy children, who are playing in the afternoon, but by the next morning, having developed malaria, are already in coma, from which many of them will never recover and will die from it.
That's crazy. So how does malaria act once it's in the human body?
So malaria is caused by a parasite that's called Plasmodium. And this is a parasite that is transmitted to us by mosquito bites. So mosquitoes are the carriers of this disease. And it's a very specific kind of mosquito known as Anopheles. And the Anopheles female mosquito likes to feed on blood because it helps with the development of the mosquito eggs within the female. But in taking that blood meal by biting humans, for example, the mosquito itself becomes infected with Plasmodium that's in the blood of humans. And then that infected mosquito can pass along that infection to the next person it bites. The parasites are in the mosquito saliva and are released as the mosquito is biting the individual looking for a blood vessel. From the mosquito saliva, the parasites make their way to the liver. They infect cells in the liver and develop within those liver cells for about the next week. In this time, the person has no symptoms, so there's no way for them to know that they are infected. So the parasite is sort of very stealthily increasing in numbers during this week. And once the development in the liver is complete, the parasites enter the bloodstream and infect red blood cells in the bloodstream. And this is when the symptoms of malaria are manifested. So this is when the chills and the fevers that I had mentioned to you earlier start to manifest themselves. And that is the first, usually sort of the first indication that the person is sick, possibly with malaria. within the red blood cell, the parasite continues to divide and replicate and increase in numbers. It will burst out of the infected red blood cell, find fresh red blood cells to infect, and just the cycle can keep going on indefinitely unless the person is treated with anti-malarial drugs. And also in this time, if this when the parasite is in the blood, if this person is bitten by a mosquito, then the mosquito will become infected and be capable of passing along the infection to another individual.
Wait. Okay, the last thing you said malaria can be passed on from individual to individual?
Via a mosquito bite.
Okay. I thought it was some other method.
It's not, so it's not, malaria is not transmitted from person to person just by contact between people. But if an infected person is bit by a mosquito, and then that mosquito goes and bites another person, then that other person can become infected. But just those two people meeting each other, shaking hands, sitting next to each other, not socially distanced, that will not cause malaria. So hope that is clear.
Yeah, no, that makes sense. Of the people that get malaria, are children most affected? Are some just totally fine? Or does it happen more in adults? Like, what's the distribution of the effects?
So the worst, 80% of deaths from malaria are in children below the age of 5. And we think that's because these very young children have not had, their immune systems have not had time to develop antibodies against the parasite and develop an immune response against the parasite. So they are very vulnerable. As you get older, if you live in a malaria endemic country, as you become older, you are just, from repeated exposure, repeated mosquito bites, your body starts to build up an immune response and develops protective antibodies. And so typically adults in a malaria endemic country, even when they become infected, don't develop the worst, the most severe forms of disease typically. They might have fevers, they might have chills, they might feel really fatigued. They will definitely feel sick, but they are far less likely to die from the disease, unlike very young children who have a much greater risk of dying from it. Another patient population that is particularly vulnerable are pregnant women. Women that are pregnant are at high risk of contracting malaria. And so that is another patient population that is really vulnerable, even though they are. Does that answer your question?
Yeah, it does. How has the way or the prevalence of malaria infection changed, in countries that are actively trying to stamp out these mosquitoes? And I figure every country on Earth is trying to do something to get rid of malaria, but what's happening? What are the methods in which they're trying to get rid of mosquitoes, and what's the consequence? What's been happening?
So actually, the very interesting side story to the control of malaria is really the story of DDT. So I'm sure you're familiar with this insecticide. Has a really bad rap for its environmental devastation that it causes. This is what Rachel Carson wrote about in Silent Spring, for example, the effect that DDT had on the environment and on birds, for instance. But DDT was was an insecticide that was used extensively for mosquito control and malaria control. But for a short period of time, it was quite effective in reaching those goals in Asia and Africa. But then its use was discontinued for the environmental impact it had, and correctly so. Also, the mosquitoes started to develop resistance to DDT. And at the same time, the drugs that were being used for malaria started becoming ineffective due to the resistance the parasites were acquiring. So there was sort of this perfect storm, which led to a resurgence of malaria, even in countries that had come quite close to eliminating it. So currently, the efforts that various countries are making to eradicate malaria within their national boundaries there's a whole set of measures they have to take. Mosquito control is one of them, using bed nets, for example, to prevent mosquitoes from being able to bite people, having some sort of vector control methods using insecticides other than DDT that do not have negative environmental impacts. Also using, in some cases, anti-malarial drugs for prophylaxis against malaria. So these are the combinations of these methods are being utilized by different. In some places have been more successful than others for a variety of reasons. In the US, for instance, malaria was a significant concern even up till the 1950s and 60s in the southern parts of the US, and further south, for example, during the building of the Panama Canal, the deaths and the illness caused by malaria was very significant. And there was a whole set of public health measures that were taken, such as draining marshland, decreasing the number of places that mosquitoes could breed, just generally higher sanitation, probably air conditioning so people weren't outside as much and more vulnerable to mosquito bites. All of these things together helped in eradicating malaria in the US. And some similar measures are being tried currently in countries that still have malaria.
Your study in particular, is it the mechanisms of transmission? Is it the biology of the parasite? Like, what are you focusing on?
So I focus on the life cycle of the parasite that occurs in the liver. So this is the very first step of a human infection, and it is absolutely essential for the parasite to be successful at this step, because this is what allows the parasite to establish a niche in the human host from which it can sort of perpetuate itself and cause the pathology and symptoms associated with malaria. So what I'm interested in learning is what are the mechanisms that the parasite uses to enter, to invade a liver cell, to develop within the liver cell, and then to exit the liver cell. And the idea being, if we can understand these steps in greater detail, then we might identify the weaker links of this cycle, and then we can target those weak spots with drugs such that the disease never progresses to the point where it causes illness. So
is the liver where the malaria will go first, or
Exactly. So the liver is the very first step in human infection. After the parasite has been introduced via a mosquito bite, it will go to the liver. And if you can prevent it from infecting the liver, or block, even after infection of the liver, if you can block it from replicating within the liver, then you have caught it at what's a bottleneck in its life cycle. At this point, parasite numbers in the human are still low, and the parasite has not increased in numbers exponentially as it will do once it reaches the blood. So this is the weak link in its life cycle in the human host.
So once it reaches the liver, what form is it in, and what does it do in the liver?
So when it reaches the liver, it is in a form that we call sporozoites. And sporozoites they're sort of like crescent shaped, or a shape, or in the shape of a banana, you can imagine. They're highly motile. If you look at them under a microscope, you see them moving round and round in circles, like moving really fast. You'd be astonished at how quickly they move. In the body, what that translates to is that the parasite moves from the skin, which is where it's deposited by a mosquito bite. That's why we scratch when we get bit by mosquitoes, because the skin is irritated. The parasite has to move from the skin, make its way from the skin all the way to the liver. So that's quite a long journey. And sporozoites are really adept at this journey. So they make this journey from the skin into a blood vessel, and then the blood carries them to the liver. Somehow the parasite realizes that they've reached the liver, and at that point they leave the bloodstream and start to infect liver cells.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Once they're inside a liver cell, they change completely in shape, completely in form. It's almost like a metamorphosis, as a butterfly larva changes into a beautiful adult butterfly, and it's hard to imagine that the adult butterfly came from this pupa. They look completely different. So the parasite does this similar kind of morphological change. It stops being motile. It doesn't move around anymore. It becomes, it loses its crescent shape and becomes more like a ball shape or a spherical shape. And then it starts to divide, goes from 1 to 2 to 4 to 16, and goes on like this for almost a week inside a single liver cell. And so this is what allows it to increase in numbers from a single sporozoite infecting a single liver cell will in 1 week give rise to tens, if not hundreds of thousands of more parasites, which are now infectious to red blood cells.
Wait, so inside a liver cell, it's been observed that the parasite will multiply until it literally consumes the entire cell and then bursts out? Exactly,
and it bursts out of the liver cell. And actually bursting out is not, it's almost, I don't have a good word to describe how it exits the liver cell, but it's not like an explosion of the liver cell. It's more like the parasite is bundled. It exits the liver cell in a very clever way. It masks itself by wrapping around itself the membrane of the liver cell itself, so that the body's defenses don't necessarily recognize it as being foreign, as being a pathogen, because the membrane, it envelops itself in a membrane that looks like the host cell membrane. And sort of cloaked in this fashion, the parasites leave the infected hepatocyte and once again enter the bloodstream. Within the bloodstream, this membrane around them falls apart and the parasites are released into the blood, and now they are free to infect red blood cells.
Huh. Well, is there an average number of parasites that will come out of a liver cell once they exhaust it?
It's been I don't I'm not sure I can give you a firm number. It's been just because this step is really hard to visualize and study intensively, but what the estimate is a single infected liver cell can release 10 to 100,000 infected parasites. And the even more amazing part is all of these came from a single sporozoite. So the logarithmic increase in numbers of the parasite in such a short amount of time, it's really mind-boggling. And that's why...
Has that been observed? Have animals, for instance, been infected with one sporozoite, and you've seen it literally multiply into billions of...
Exactly. If they're infected with a single sporozoite, it will divide to form something like 10,000 to 100,000 liver stage parasites. And each of those parasites, when it infects a red blood cell, will divide 16 to 32 times more. And so it goes from 1 to 10,000 to 100,000 to a million, all in the course of 1 to 2 weeks.
What's the immune response from people?
Right. So
how does it hide?
So people, our immune system does recognize the parasite. The immune system does understand that this is not a good sign. But the it's really kind of an arms race. It's really the parasite trying to keep one step ahead of the immune response. So, for instance, when it's in the liver, the immune system, of course, it takes some time for the immune system to recognize that there is an infection. Then the immune system has the problem of trying to find the one infected liver cell amongst millions of uninfected liver cells. So it's really a problem of looking for a needle in a haystack. And all of this time, the parasite is dividing in the liver and getting ready to leave the liver. So the immune system is always playing catch up. So if the immune cells do not find the infected hepatocyte in time, then the parasite can complete its development cycle in the hepatocyte, which is the cell in the liver that is infected. By the time the immune system catches up to the infected liver cell, the parasites are already out of the liver and into the bloodstream now. So now the immune system has to start identifying infected red blood cells. And so it starts again from scratch. The whole time the parasite is really good at changing its surface coat of proteins. So there is a lot of what the term we use, antigenic variation. And it's as if the parasite is continually changing the coat it has. And sometimes the coat is of one flavor, another time it's another flavor. And the immune system, by the time it catches up to one flavor and is effective against it, some of the parasites have already changed the coat and they escape that immune response. And so there's always, the parasite tries to keep one step ahead of the immune system, and the immune system is playing catch up by producing antibodies against the parasite, producing T cells that recognize infected hepatocytes. And sometimes, for example, in the case, for instance, of coma, which is when kids die of malaria commonly, the immune reaction itself can be harmful and lead to the pathology that causes. So that is what the immune reaction is doing in the case.
Has anyone, so malaria, it affects people, but does it affect animals too? Can you do animal studies with rats or with monkeys, or what do you have to use?
That's a really good question. So the species of Plasmodium parasites that infect humans in general do not infect other animals, and vice versa, meaning species of Plasmodium that infect other animals, and there are species of Plasmodium that infect birds, for example. Plasmodium infection is particularly bad for penguins. Penguins do not have a natural defense against malaria because typically in the Antarctic, penguins do not see Plasmodium. But when you have penguins in zoos or captive penguins that are being rescued from oil spills, for example, then those penguins come in contact with species of Plasmodium that infect other birds, such as pigeons and other things that are flying around. And penguins can become infected with Plasmodium, and it can be really bad, and it can kill penguins. There are species, but that species that infects penguins and birds does not infect humans. The species that infects rats and mice does not infect humans. And similarly, the species that infects humans does not infect penguins or rats and mice. So there is species specificity. There is a species of Plasmodium that infects primates, monkeys, and can also infect humans that come in contact with. So that is a zoonotic species of Plasmodium. But mostly Plasmodium species that infect humans are specific to humans. Other animals also get Plasmodium infection, but they don't necessarily have disease in the way that we think of malaria in humans.
Well, are you able, have scientists done biopsies and removed one or more parasites and then studied them?
Do you mean have they removed parasites from other animals?
From a person to study them.
You can certainly get red blood cells from infected humans, and this is collected in the field quite frequently. We can study parasites obtained from human patients in that way.
Well, what's been noticed? What kind of learnings can you get by doing that?
So for one example, one thing that's very important for us to understand what's happening in the field is in order to understand resistance to anti-malarial drugs. So parasites that are found commonly in the Thai, Cambodia, Laos region in Southeast Asia, parasites that infect humans in that region of the world have become at increasing frequency show resistance to drugs that are used against malaria now. So these are drugs known as that combinations including artemisinin and other drugs together with artemisinin. And the parasites that we obtain from human patients in the Thai, Myanmar, Laos border area in Southeast Asia, these parasites often show resistance to the artemisinin combination therapy. So by collecting these parasites, we can monitor how much resistance there is, how quickly drug resistance is spreading through the population. This allows clinicians to adjust the dosing, the doses that they give their patients, and in some cases change the drugs that they're using altogether. So that is one example of how using parasites collected from human patients in the field can be.
Has anyone looked to see if there's a microbiome of the parasite, and if has anyone captured it in the liver and looked at it, maybe looked at its microbiome, and then captured it, in a red blood cell and see the changes?
So I think I'm not sure about that the parasite itself has its own microbiome, because the parasite lives within a human cell, whether it be a liver cell or a red blood cell. But I think it's an interesting question to see how the microbiome of the human patient might affect the disease or the progress of the disease, because we know that our microbiome, the microbiome in the gut of humans affects our immune system, and that effect on the immune system could translate into an effect on how well malaria infection is controlled, or might affect the immune response that individuals mount against malaria. What people have done by obtaining parasites from an infected liver cell or infected red blood cell, certainly other researchers have examined what are the genes that the parasite expresses in these stages, what are the different proteins it makes, and even how it alters the cell that it's residing in. So how does it change the liver cell that it's growing within? How does it change the red blood cell that it grows within?
Morphologically or function-wise, when you compare the parasite when it's in a liver cell versus a blood cell, how different is it?
So in some ways it is similar, but in other ways it is quite different. One big difference between we see in an infected red blood cell compared to an uninfected red blood cell is that the infected red blood cells are often deformed. There are sort of knob-shaped structures on the surface of an infected red blood cell, and these knob-shaped structures consist of proteins that the parasite exports to the red blood cell, and they change the structure of the infected red blood cell, making the red blood cells more sticky so that they don't circulate in the bloodstream as well, and that saves them from being identified as infected cells within the spleen and being cleared out. So it's like a self-preservation strategy on the part of the parasite. Similarly, in the liver cell as well, the parasite makes changes to the liver cell so the parasite can acquire nutrients from the hepatocyte that it needs for its own growth. And some of the pathways that the parasite uses in the liver and in the red blood cell are different. So we see that maybe the need for fatty acids is higher in the liver stage parasites than it is in the blood stage parasite, for example. So the blood stage parasite needs to break down hemoglobin, because hemoglobin is the major component, is a big part of a red blood cell. But the hepatocyte, on the other hand, has no hemoglobin, and liver stage parasites don't need to break down, don't break that down. So that's another difference. There are lots of differences between the two stages, and some similarities as well.
Huh. Well, I didn't ask you, once it's in the red blood cells, then what?
So within the red blood cell, again, the name of the game is to divide, increase in numbers, exit the liver cell, find a new cell to invade. So the word parasite itself means it cannot live independently. It always has to be within a host cell. And so the parasite, once it's within the red blood cell, it divides to, a single parasite will divide to form anywhere from 16 to 32 new parasites. These parasites will explode out of the red blood cell and find new red blood cells to infect. And so this is the reason that this continual invasion and lysis of red blood cells is the reason that malaria patients develop severe anemia, because their red blood cells are being constantly eaten up by the parasite within them. Inside the red blood cell, the parasite continues its differentiation cycle. And also, if it can get taken up by another mosquito, that's really good for the parasite as well, because that is what enables the parasite to spread to another human.
So at the point it's in someone's blood, that's when the parasite's hoping that a mosquito will bite that person.
Exactly, because when it's inside the liver, it really can't be taken up by a mosquito. But once it is in the blood, then it is accessible to a mosquito. And because when the mosquito sucks up blood, it's also going to suck up some parasites that are in that blood.
Well, people that have malaria in their blood, are they symptomatic at this point? Or do they feel fine still early on?
So I think it depends on your immune status. So if you are someone who has lived in a malarial endemic country, then you can have a low number of parasites in your blood, but not experience any overt signs of it. On the other hand, if let's say you are a traveler who has gone from a country with no malaria to a country with malaria and have become infected there, and this is your first time becoming infected, then even with very few parasites in the blood, you might develop quite severe disease. So I would say that there is a range of symptoms depending on what your previous exposure to the parasite is.
Well, what changes inside someone once the parasite's in their blood? Do they become more? Do they tend to get bitten by mosquitoes more often? Do they give off, all of a sudden a pheromone that attracts mosquitoes more often? Does the parasite cause that to happen? Or
that's a really good question, and there is some suggestion that might happen, that the parasites, release some sort of chemical, some sort of signal that can be picked up by a mosquito and sort of signal to the mosquito that, that's attract the mosquito to that person. It's not, that work is really very intriguing, but we still need to see more to be sure that happens in real life. Certainly, as you probably know from experience, there are some people who seem to be bitten, that they seem to attract mosquitoes so much more than others.
Yep.
And that is thought to be because of, smells and odors and chemicals that certain people release more of than others. And if these are chemicals and odors that are attractive to mosquitoes, then those people get bit by mosquitoes more often, regardless of whether they are infected or not.
Yeah, no, that's true. Has anyone identified what makes someone juicier and more likely to be eaten by a mosquito?
Yeah, there's carbon dioxide is one attractant for a mosquito. There are other chemicals as well that seem, different mosquitoes respond to different chemicals. So this is a very active area of study, but it's outside my area of expertise. So I can't, afraid I don't have the most up-to-date knowledge on that field. But there are definitely chemicals that some people release that have been identified that act as attractants for mosquitoes.
So what are you trying to figure out in particular? What's your goal?
So my goal is to understand what it takes for the parasite to successfully infect and develop within the liver, and then how we might interrupt that cycle to find drugs that will then. So that is.
Are you able to? I don't know. I guess people are not symptomatic when the parasite's in their liver, right?
Not at the moment, but there are people trying to develop diagnostic tests for this stage of infection. And also we know from the epidemiology, we know during, the rainy season, for example, the mosquitoes population increases in numbers. And so we can predict that around these months, people are likely to be bit by mosquitoes and have infection in the liver, even if they do not have symptoms yet. So we can if we have these drugs that target liver stages, we can use those drugs in a preventative way based on what we know of the epidemiology of the. So that would be the idea. Another group that would benefit from drugs that target the liver stage are travelers from non-endemic countries. And as we mentioned, there are many countries that have succeeded in eliminating malaria altogether from their borders. But people from these countries might travel to neighboring countries where malaria is still present. So those travelers could take drugs like the ones I'm talking about and that would save them from becoming infected when they go to malaria. And now, you
can't even biopsy someone.
Mm-hmm.
I guess it wouldn't even be useful to biopsy someone. Like if you knew, all right, this time of year in this area, a lot of people are very likely to have the malaria in them. But even if you tried to biopsy them, if it's just in one cell, I mean, you're not going to find it.
Exactly. So the diagnostics would have to rely on markers in the blood, for example, and not necessarily be looking for that one infected cell in the liver. But we would look for telltale signs that infected cell would be released. So those.
What about do you know anyone that's making liver organoids? Maybe you could have them test this out and see what they see.
That's a very good idea. That is a very good idea. And we've been trying to develop those systems. That would indeed be very, very.
Yeah, there's a lot of organoid people. There should be some that'll be willing to work with you on this.
Yes, it's the liver organoid people often work on human organoid, right. And then we would need human infectious sporozoites to infect those human liver organoids to study. But that would be a very useful thing to do.
I mean, even are you able to culture even, a couple of liver cells in the lab and, keep them alive for long enough and try to infect them with a malaria parasite?
Oh, absolutely. We do that routinely. We infect tissue culture, hepatocytes. In tissue culture, we infect them with sporozoites. We watch the parasites development within them. We can even, if the parasites develop completely within these liver cells, we can collect them from the extracellular medium of these cells a couple of days later. And once the parasites are in the medium, this is equivalent to them being released from the liver cell inside the human into the bloodstream. So we can collect this containing parasites and inject them into a mouse and watch the red blood cells of that mouse become infected with the parasite. So yes, absolutely, we can. Indeed, that is what we do when we screen for drugs that might prevent the parasite's infection of the liver or development with exactly the kind.
So any sense of a breakthrough that would be very helpful in combating malaria, or at least understanding it?
I think there are so many things that we need that would be so helpful. Anything, new drugs would be really useful. Understanding how the parasite, being able to diagnose the parasite in the liver would be very useful. One thing I didn't mention at all is that there is a species of Plasmodium found in South Asia, which can go into dormancy when in the liver. And then that dormant liver stage can reactivate months and sometimes years after the original infection has cleared and cause disease once again. And we have very few drugs that infect these dormant liver stages. But these dormant liver stages cause a lot of disease. Even if they don't cause death, they still cause a lot of serious disease. So if we could understand what makes some liver stages go into dormancy and then what wakes them up again, that would be a great breakthrough as well.
Yeah. You have no idea what causes them to go into dormancy?
We suspect that it's a combination of the host's immune response, but really we don't have. That's a very broad statement. We need it to be much more specific and much more precise.
Interesting. Well, very good. What's the best way for people to find out more about your work, Purnima?
They can certainly reach out to me. Anyone who'd like to know more about my work, I'm available. Email. The lab has a website that gives a very brief introduction. They can reach me by email or a phone call.
Okay. Well, very good. And last question. What do you think is going to be possible maybe in the next 5 years in terms of malaria? And what do you think is going to take quite a bit longer? But what's like the ideal that you would like to see, 15, 20 years out?
So I think the next 5 years, my answer, if we had had this conversation before COVID, my answer would have been one thing, but post-COVID, really, my answer is quite different. And at the moment, what I hope for the next 5 years is simply that we do not reverse these very hard-fought gains that we have made in malaria control over the last 10 years. Given the impact that COVID is having on health systems around the world, the impact, the attention that is, when you have such a major pandemic, it's understandable that attention shifts away from other diseases, even ones like malaria that have been around for centuries and will continue to be around with us unless we keep our eye on the ball and sort of keep pressing the gas on malaria control. So I hope in the next 5 years we continue to see improvement in malaria control, and in 15 to 20 years, really malaria is a disease like smallpox or like polio, meaning to say it is a disease that has been eradicated.
Yep. Yeah, I've heard tuberculosis, malaria, everything's, coronavirus is like a scientific parasite. It's sucking up all the attention and multiplying and taking over all the labs. Nothing else is being done. So yeah, absolutely. I understand this cannot be ignored.
Yeah, exactly. It should not be ignored because we have seen before with malaria when attention shifts up, it's a resilient disease, and as soon as attention shifts elsewhere, it is able to come right back. So I really hope the same thing does not happen again. in the context of COVID and coronavirus. And, we have no idea what co-infections with coronavirus and malaria will do patients, right? We are only beginning to see what this epidemic is doing in Africa and in India. And I just really hope that it does not make a bad situation even worse.
Very good. Okay. Well, Purnima, thank you for coming on the podcast. I appreciate it.
I enjoyed my time. Thank you very much for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.